My name is Christopher Mjawi, a master's student in the crop science department at the University of Illinois. Today in this video, I would like to share a little bit about the root digging and cleaning protocol we use in our organic controlled experiment.
We want to understand how below ground traits like root angle and root complexity change as the soil nitrogen and root competition changes, and how these below ground traits eventually correlate with the general performance of the hybrid. This evaluation is done when the maize plots start silking. This is also known as the R1 growth stage of corn. For root digging, we select the middle 2 rows in the 4-row plot, and within these rows, 3 consecutive average plants representative of the entire plot are selected and uprooted using a shovel which is oriented at least 1 foot away from the plant and at approximately 60-degree angle. Each root is then labeled using a plastic tag. It is very crucial that the labels are tied tightly using zip ties because we don't want to lose any roots. Remember, a root without a label is a data point lost. For the first 2 years we conducted this experiment, we had to load the dirty roots on a trailer and transport them to the agroengineering farm, where we immersed them in water tubs for an overnight. We also added a cleaning agent called Calgon, which seemed to help to loosen up big chunks of soil and made it easier to remove when we dunked the roots in water multiple times. We then used high-pressure water hose to thoroughly clean the roots before imaging. However, this method turned out to be very laborious, very messy, and time-consuming. Therefore, this year we tried a new method where we used the air spade to clean the roots the same day in the field, and we leave all the soil in the field. This air spade has a portable size industry compressor which uses compressed air to remove soil from the root system, leaving a very clean root almost ready for imaging. The air spade method seemed very effective and conserved most of the root structure due to less manipulation of the root system compared to the previous method we used in the past 2 years. The cleaned roots were then immersed in water tubs at the agroengineering farm to thoroughly remove the remaining soil before imaging. The root imaging was done using a corn root imaging box, which is a high-throughput root phenotyping instrument developed by Dr. Tony Griffith here at the University of Illinois. The root is inserted in the crib and images are taken by the top view camera and the side view camera as it rotates on the spike. These images are later analyzed using a programming software called MATLAB to acquire phenotypic data describing the maize root system. When we look at the preliminary analysis we did using the data from the first year of this experiment, it showed that in general, increasing soil nitrogen levels significantly reduced the complexity of root systems, but increased the root angles. However, some of the hybrids showed this response more prominently than others, depending on the breeding program they were developed from. For now, this is what we know. However, more conclusions will be drawn when we combine our data from the past 3 years and conduct a detailed statistical analysis. These results will be available to you very soon on our website. Thank you.